Oh, and as Gail said, the session is being recorded today, and will be distributed to all participants afterwards. So, yeah, if you do happen to miss something, you can catch up on the recording afterwards. So I think that's pretty much our 2 minutes, and hopefully everyone that's looking to dial in has dialed in, but anybody that hasn't will just catch up as the session goes live. So I will click through. So yeah, thank you for joining today. This session today is on the CMC development of new small molecules, new molecule products. The panelists for today. So my name is James Crooks. I'm the Staffing and Performance Director at Product Life Group for Regulatory Affairs. We have expert panelists today who are Rania Jalal and Fabio Pedner. So myself, as I've mentioned, I'm the Staffing and Performance Director. I've got 20 years of experience in the pharmaceutical industry, with the last 10 years focused on operational delivery and senior management within regulatory affairs. The Staffing and Performance Director and Global Regulatory Director at Product Life Group. I have expertise in capacity and capability management, proactively addressing resource constraints, and directly supporting global staffing strategies. So I play a key role in recruitment and workforce planning, leveraging Product Life Group's global capabilities to meet our business demands and drive growth. And really part of my role is also identifying expertise and where our specialist subject matter experts sit within the group. And that really leads me on to Rania and Fabio. So Rania is a senior regulatory CMC consultant. She has 20 years of pharmaceutical experience, primarily in the CMC space, with 12 years in regulatory CMC supporting different size organizations. Her focus has been on product development for small molecules and drug device combination products across various dosage forms and routes of administration. In this webinar, she'll be speaking to the CMC development of small molecules and to the regulatory CMC aspects related to their development. Also supporting her today in the webinar is Fabio Pedner. He's our CMC service area manager within Product Life Group. Fabio has 14 years of experience in regulatory CMC, primarily focused on the development of new pharmaceutical products. His expertise lies in CMC development for small molecules across various dosage forms and routes of administration, including authoring and driving CMC dossiers for clinical trial submissions and marketing applications, as well as briefing packages for scientific advice. In the webinar, he'll be focusing and discussing on the key challenges encountered during CMC development and sharing real-world examples to illustrate practical solutions. So that's our panelists for today. So I'm really pleased to introduce them, and I will be merely moving through the slides for this session and also hosting a Q&A session at the end. So for today's agenda, we'll have a brief introduction, giving an overview of CMC development for small molecules. In section 2, we'll look at the CMC as the foundation of quality development and look at that CMC role across all phases and also touch a bit upon quality by design. As we move through, we get to section 3 on the critical CMC challenges, looking at things such as starting materials, impurity control, dissolution and briefing strategies, and moving then through a view on the future perspectives in CMC, where we will discuss around continuous manufacturing, real-time release, and also the introduction of artificial intelligence in regulatory affairs. We will wrap up with a bit of a conclusion and provide some speaker insights and a summary over the entire session before landing on the Q&A session. And as Gail said at the start, there is a chat box that's open. You can add your questions and answers throughout the session. We will look to compile those throughout the session, and then I will pick a few select ones at the end and then fire those towards our panelists. So hopefully we should have about 5 or 10 minutes at the end of the session to really go through those. So without further ado, I will hand over to Rania, who's going to be doing the introduction. So thank you, Rania.
It's me, James.
Oh, sorry, my mistake.
Yeah, don't worry. Okay, so thank you.
Live show's gone slightly wrong at the start, but yeah, Fabio with the introduction. Apologies.
Thank you very much. You can go also in the next slide. Thank you very much, James, and really hello to everybody. So it's a pleasure for me to be here to present this webinar and to talk about the CMC role into the development. And I hope my experience will help you to navigate in this complex topic. And we are choosing this topic because we really deem that the CMC role in the development is crucial, but it's a role that it's often also misunderstood or underestimated, because in my experience, at least, there is the general perception, no, that the CMC comes into play only when the commercial manufacturing takes place, which is really not the case, because there are so many quality challenges during the development in which the CMC can step in and provide an added value. And so this is exactly what we want to do today, to show you how the CMC can step in, which is also the method that we should apply here to really support in an efficient way the project team. And let me be also clear on what will be out of scope in this webinar. So we will not talk about the large molecules biological products. And also, it's not going to be a deep diving in the content of the ICH regulation or country-specific requirements, because we want to set up this more CMC strategy discussion, where we will show a method that you can then apply across different countries and situations. Okay, so say that, really hope you will enjoy the webinar, and I will leave the floor now to my colleague Rania for the first part.
Thank you, Fabio, and thanks, James, for the introduction. I think we can move to the next slide. So these are, for lack of a better word, what I would call more of like the pillars of CMC, or chemistry, manufacturing, and control. And so they present really the essential elements required to ensure the product is safe, effective, and consistent from early stage through commercialization. CMC is really key to successful drug development. And the CMC section, which is known as Module 3, is critical components in clinical applications and also marketing applications, so regulatory submissions. So in essence, IND, IMPD, 505B1, B2, MAA. So the data provided in the CMC section within Module 3 should provide the health authority information on what you see here, which is the drug's identity and structure, how it's made, how it's controlled to ensure the quality, the safety, the potency, how stable it is, and really how pure it is. So what potential safety risks may arise from impurities, from drugs. The ultimate goal and responsibility of the applicant or the sponsor is to ensure the drug is safe and effective for the proposed use. Obviously for the proposed use, and that the benefits outweigh the risk. So in essence, the labeling is appropriate, contains all the necessary information. The manufacturing section, the manufacturing method, preserve the product identity, the strength, the quality, and purity. Next slide, please. So how is the product life cycle changing, and why is the product life cycle changing? So there's an increase in developing, as we more and more understand the diseases, and we go toward targeted therapies, and we have more of these orphan drugs and orphan designation and fast track designation, accelerated pathways. More and more sponsors are using the accelerated pathway. So the nature and challenges of the orphan drug development inherently lead to frequent and complex CMC consideration throughout the product life cycle. Now, this is great for patients, but it's terrifying for CMC, because this really means that the CMC change is constant. When you're thinking, for example, of orphan drugs, once you think of orphan drugs, you're thinking of aggressive timelines, right? You want to get to the market ASAP, unmet need, accelerated timelines, expedited regulatory pathway. So that really compresses the development timeline. This means that the CMC must always be very proactive, thinking 10 steps ahead, There's a lot that will be, the activities that will be pulled forward, conducted at risk. sometimes even before clinical results can fully guide the process optimization. There is, in some cases, limited process data at launch. So, you want to achieve to get to the market faster. So let's optimize the manufacturing process compared to traditional drug and the traditional pathway, right? Sometimes that's why the agencies sometimes work really well with the industry pharma. They cut you a slack. The formulation is complex. The manufacturing itself is complex. A lot of them, for example, are injectables. So solving for these complex chemistry problems. And then, of course, being orphan, for example, it means small, high volume batches. So, you're working with a small patient population, that would require sometimes changes in demands, manufacturing flexibility. And that leads to more frequent adjustment to the batch sizes, the equipment, we're changing from one manufacturing to another. So in essence, all that requires sound science and also regulatory flexibility. There's a lot of gray area. There isn't something as simple as I'm going to look at the guidance and follow it. It's the case by case. So the move toward orphan drugs with their inherent complexity and expedited pathway, it creates that environment where adaptability and continuous change management and CMC is essential for the product development and the success of the program. We can move to the next slide. Thank you. So this is more or less just of kind of like a simple visualization of the CMC role across the product development. So we're going to be discussing the regulatory CMC strategy at different phases of the drug. This slide kind of sets the stage for our discussion. So, as you can see, it shows the stages of development from phase 1 to approval. Note also here what I have in the orange, which is the major FDA meeting opportunities at the top. They're not inclusive. They're not all inclusive, but you'll see pre-IND. You'll see, end of phase 1, there's end of phase 2, the pre-NDA or pre-MAA. And of course, there's other opportunities like type C, type B. In essence, this is where the sponsor has the opportunity to prepare the briefing packages, meeting requests, and pose the proper questions to get the best feedback from the agency, align, right, on the CMC plan and CMC activities. The text below that, right below the orange, that kind of shows the typical CMC changes that occur during development. So changes to scale, process, we're changing manufacturing site, we're changing the testing site. We're at a phase 2 and we're changing the container closure, changes in the formulation. The text at the bottom all the way, that it kind of shows the status of some of the major CMC activities. Obviously, that's not an exhaustive list, but it just kind of gives an example. And as you can see, you start thinking of specification early on. Now, as you evolve and understand your product more, it becomes not just, hey, I need to have the following spec. It's, I need to tighten them. I need to revise them. I need to understand them. I need to justify them. So it becomes much more comprehensive. And a lot of justification is, required as you further understand the process. So well-defined spec, let's say, is validated, etc. Throughout the whole development, the major question, as you're early in phase 1 that's on the mind of the health authority is the product safe to administer to a small number of human subjects? So safety, right? As development progress and toward late phase, the health authority will continue to focus on, the product safety, but it's more also on in large group of human subject. And it's also the focus becomes on how does the package look like? How does module 3 look like? What is the story we're telling? So since CMC changes are constant throughout the product life cycle, the sponsor need to ensure they have the information needed to bridge the batches used to obtain safety information to the proposed clinical batch, and really throughout development, consistently demonstrating equivalency between batches is critical. Continuing to ask if there's a clear bridge to the final product, ensure an efficient path. Changes are expected, right? And the health authority understand that. It's all about, are we showing bridging? Are we showing equivalency? That's really the key here. And as you can see, the last thing I have in this slide is that kind of big arrow QBD. And that is just to say that the quality by design or QBD approach has to align with the product and process development stages and clinical development phases. It is not something you apply at the end or you apply at one phase. It is something you apply throughout the cycle of the product life cycle. Next slide. So, we just said QBD. So just kind of, I'm sure a lot of people are very familiar with what QBD is. So this is, again, more or less just of a visual. Again, as we said, it aligns with the product and process development stages and the clinical development phases. QBD is more or less of a systemic approach to development, and it applies to early phase, it applies to late phase, it continue to apply to the product life cycle. So it applies to IND, NDA, etc. It begins with predefined objectives, and that's why usually when a program gets launched, the team from all cross functions meet. They define what is the target product profile, what's the desirable, what's the minimum, and then from there they generate the CQA. Well, that's the beginning of the elements of the QBD. So applying and trying to understand as much as possible throughout the product life cycle. It's based on sound science, and it's based on quality risk management as well. Failure to apply QBD is not going to give you shortcuts at all. It may sound like it's making the approach longer at the beginning, but it really pays off because it enhances the control strategy. It complies with the agency expectations, so less questions from the agencies that you can't answer. It improves the change control and the lifecycle management, and it prevents manufacturing failures as well. So you're really showing the agency that you understand the upstream, you understand your product, you understand the variability of your process, etc. How to use the elements is really at the applicant discretion. Typically, at the beginning of the phase one, when you're thinking QBD, you're focusing really on the product understanding. Phase two is you're focusing on the process understanding. Phase three, you're applying everything your product, your process to manufacture your phase three and what we call your primary stability batches or registration batches. Next slide. Thank you. So CMC role, and really here when I say CMC, it's CMC and Reg CMC, as they have to work together. So this is, let's explore it during early phase of product development and see how it evolves. So the early phases, the primary focus is ensuring I have sufficient CMC data to assess the safety of the proposed clinical. So the agency understands, the agency body, that the manufacturing is still in development, it's not fully optimized, it's not validated. The focus really at the beginning is just making sure whatever data we have is going to support the safety of the product. So it's allowing the clinical trial to proceed without delays. We don't want a clinical hold. One of the challenges, because that occurs usually when you're thinking of, like filing for an IND or an IMPD clinical applications is, one may think, oh, well, we're in early phase, so it's kind of straightforward, less data. But there's actually a bit of challenge in it because when you look at the guidances, they're really made, most of the guidances set for marketing applications, right? There's a little bit that talks about early stage. And so it's really the role of the sponsor to interpret and determine the appropriate level of information and control for their current phase of development. And that is different from one modality to another. If you have a solid dosage form, that's different than an injectable. That's different than a cross-labeled combination product. So examples on, information to think of and ensure you include is the impurity profile, stability data, how much stability data. And obviously for, early clinical, the important thing and the agency want to make sure is you're clear on the length of your clinical study and you're explaining to them and you're clear that your product will be on stability support and monitor throughout the duration of the clinical study. So whether you're pursuing a 505(b)(2), a 505(b)(1), an MAA, early engagement with the agency, so pre-IND, end of phase one, is very useful to get specific feedback on the sponsor's proposed CMC strategy to avoid potential clinical hold later in development. And as we go further in development and more information is obtained, it becomes even more critical to have these meetings with the agencies. So try as much as possible to always engage with the agency and get alignment early on. Next slide, please. So we're still talking about early development, since we talked about, the guidance, not enough guidances, to tell us what to include, what not to include, and it's a case by case. Early development starts with IND-enabling studies. So those are basically, assessing the new chemical entity through performing a set of pharmacological and tox studies on animal species. So GLP studies are directly connected to the drug product. This means any changes in the drug product after you perform what we call the preclinical batch or the tox would require bridging. So going back and talking about the bridging and showing equivalency between the preclinical and the proposed clinical. In fact, the FDA did refuse to allow clinical protocols to proceed when differences existed between preclinical and the intended phase 1. So again, initiating early phase conversations, early FDA conversation, early conversation with the EMA, etc., to have a clear understanding of the regulatory requirements for your specific product is really essential. Next slide. So how does it shift and how does it evolve as we go toward phase 2 and phase 3? And, in phase 3, now we are preparing really for thinking as much as possible, what is my story going to be? How is it going to look like? What is going to go in the submission and what is going to be post-approval? Am I pursuing this path or that path? What is my short-term plan? What is my long-term plan? There's a lot of complexity. There's a lot of strategy. There's a lot of parallel path. There's a lot of changes. How do I maneuver that? How do I communicate to the agency? In around that phase, let's say phase 3, your manufacturing process should be well defined. The process should be, the sponsor should be using a process and facility that can be scaled up for commercial production or a plan to bridge one. So if it's not, there has to be a plan to bridge one. This is where now the specifications, it has to be very clear to the health authority that we really know what we're talking about. So the specifications has to be tighter. They have to be justified. Our critical quality attributes now are very well understood. Our analytical methods are validated. Again, where it comes to case by case, if it's an injectable, a sterile product, full sterilization process validation would need to be submitted. Stability becomes not just, hey, I have some limited stability to support the clinical trial. It is a comprehensive stability program, full understanding of this is kind of what I'm going to be doing to understand the shelf life of my product, the recommended storage conditions, etc. And so this is also the stage where it's very important to have your end of phase 2 meetings. Am I going to have an end of phase 2 specifically CMC meeting with the agency, pre-NDA, pre-MAA? Do I need a type C meeting, etc.? Next slide. And so as we move toward the submission, now we're shifting from as simple as, I have enough supplies to support the clinical trials to how is my package looking like? Do I have all the validated data? Do I have my story locked in? How am I going to be telling my story? I am not going to undershare, but I'm also not going to overshare. Everything you put in your package, you have to also think, what is the agency going to ask? How is the agency going to interpret it? So, you will have to be generating your gap analysis, risk register, what we call a risk register, meeting as a cross-functional team and talking about these are the possible risks. Here is how we're going to mitigate them. If the agency asks for this, we're going to provide this. So, the risk register is not something that starts late, but this is where you have more and more information to actually sit as a team and have a proper risk mitigation. The agency expects that, by that time, your process is well defined, the product is fully characterized, and of course, it aligns and, comply with the ICH's requirements, so Q7, Q8, etc. During the preparation of the marketing application, Reg CMC really plays a huge role with CMC. Sometimes CMC subject matter experts are, extremely technical. They really know the process very well, understand the product. But telling the story in Module 3 is an art. And so that's where Reg CMC comes in to ensure that the story is told in a very compelling, way, a very coherent way, as much as possible, preventing unnecessary questions from the agency. So this is really just kind of high level to what the CMC challenges are throughout the product developments. again, as we said, and we showed in that visual presentation, the alignment of CMC and clinical development is really essential. Sometimes a company may inherit a product, and a program, and that program, for example, could be at a phase 2 or a phase 3. And then, they receive the CMC package with that, and the CMC is lacking. It's barely like a phase 1. So there's a lot of, how do we ensure that our CMC align with the clinical development? Again, talking about when we're talking about expedited and accelerated programs, that's where direct CMC and CMC, it falls on them to ensure that with sound science, we are really approaching a risk-based approach, because some risks will have to be taken to get to the market as fast as possible. So again, this is where proactive communication with the agency is really, really important. And again, as we talked about QBD, applying QBD early on throughout development, implementing the proper elements as we go would really ensure a more scientific and risk-based approach to the regulatory oversight. So I think this kind of leads us to more real-life examples that Fabio will be presenting to us on critical CMC challenges at different phases of the drug product development.
Yes, thank you, Rania. Really, I think you have very well explained how important is the role of CMC into development, how it is spread across all the different phases, and more important, how is really key to have the right method to properly support the team. Now I will deep dive a little bit to show you how you can really practically implement this method in some of the most common and also critical challenges that the CMC used to face during the registration procedure, but also during the development part. So if you go to the next slide, we can start from a quite big one, that is the definition of the drug substance starting material. So this is important because this is really the core of the development of the synthesis method for the API. And we know that the ICH regulation defines criteria for the starting materials, but does not provide so exact and precise indication that you can apply to each and every case. But it's always a case-by-case evaluation that needs to be done based on your molecule, on your process. And it's always a sort of balance game where the applicants want the starting material to be defined as close as possible to the API to reduce the number of GMP steps and to simplify the control strategy. But the assessor often challenge this because wants the starting material to be moved more upstream to control in the synthesis process. And so how can we be sure to convince the assessor that our justification is scientifically sound and will be accepted? And this is where CMC can play a role because applying the method in this discussion, so the CMC can help to define which are the critical quality attributes of the starting materials, and then to see how the quality of the starting materials it's impacting all the downstream steps and so all the intermediates up to the final drug substance. So this is very important, and this can be done in all the critical quality attributes of the starting material, but especially I would say for purity, because purity is key for the synthesis process. And so it is you need to identify both the potential and actual impurities of your starting materials, and then challenge your synthetic process on how it is effectively capable of purging those compounds in downstream steps. And so you can do this by a theoretical evaluation, and then sometimes you can also you need also to do spiking experiment to confirm the purging capability. So if you do all this risk assessment process, then you end up to set specs for the starting materials, for the intermediates, and for the final drug substance that are not based only on the actual results on your final batches, but are really part this overall risk assessment process and all interconnected one to each other, and so you really show how you have a proper control on the process, and this is really what they are expecting to see. So and to obtain this, so the CMC plays a crucial role because need to connect with scientists, with the labs already during early stage of development, and to make sure this is applied. And also it's encouraged to have, as Rania was saying, some preliminary meetings with the health authorities for scientific advice meetings to have their preliminary feedback on the justification. And so usually the CMC is the function that prepare the briefing book package for these important meetings. If you go to the next slide, indeed, I have prepared an example. Yeah, you can click on the next slide. I prepared an example on an extract from a briefing book for which I have worked recently to support a client that touches really all the points that we have discussed. So I will not go into the detail of all now because I don't want to repeat myself, but really it touches all the points that we discussed in terms of approach. And I suggest if you have time when you will have the slide to have a look on this, because it's always useful to see a practical implementation of this theoretical assessment. And just to show you that these have been really accepted very well by the health authority that has provided a positive feedback. Okay, so I think, James, you can skip the next couple of slides. Yes. Gain. And this brings me to the next challenge, which is the definition of an impurity control strategy that is really very important. We have touched this point already during the starting material discussion, but of course it's not only important for the starting materials, but also for the drug substance in general, but even for the drug product. So it's a general topic. The assessor are really posing a lot of attention in requesting from the applicants a strong and robust control of the impurities, so a really robust strategy, because at the end it could be a potential safety issue. And so it's really important. And so how can we be sure to meet the assessor expectations? So to be very clear, it's not sufficient to just base the control strategy on the results of the final batches, on the actual results, and so on the impurities that shown above the reporting threshold or the ID in your final batch. This is not sufficient because you need really to start from the beginning and to start from a risk assessment process with in mind the understanding of your molecules, of your process, the pharmaceutical forms, and all the criticism that could exist. For example, some weak bonds in your molecules, or some steps in the process with high temperature, with strong condition that could raise the formation of impurities. And so with this exercise, then you end up by defining a quite extensive list, in my experience, a list of potential impurities that could be theoretically present in your process. And then in the results of your actual batches, you may see only a few of those impurities really present above the reporting threshold, but you still have to act also towards all the other potential impurity that result from the risk assessment. And you need to challenge your process if it is really capable of purging those compounds during the synthesis or the process. And also in terms of toxicity, is requested that you assess toxicity of all the potential impurities. So this is the process, and this is done usually by a stepwise process. So you start with theoretical evaluation. For example, you can assign a sort of purging factor to all these potential impurities that estimate the capability of your process to effectively purge those compounds in the following step in the process to exclude any risk of carryover to the final product. And then if this purging factor is exceeding the threshold, then you want to test also from using spiking experiments to really confirm that your process is able to purge the impurity. And if not, then you need to take corrective action. So, and even for toxicity, you start with an in silico evaluation theoretically. Then if you have some alert, then you do the AMS testing to confirm if there is any toxicological risk. And if yes, then you do a toxicological study. So this is the process, and I can't stress enough how the CMC role is important in this, because it needs to connect with the scientists already from the early stages to set the process and to also prepare briefing book packages for scientific advice. Even in this case, it's really encouraged to have this kind of meetings, and CMC is leading that process. If you go on the next slide, please, there is a quite nice example of an activity for which I've been working with a client recently, and it's really an example of how this, an application of this process, because there, from the risk assessment, one of these potential impurities, we realized that there was a real risk of having a carryover in the final drug substance. And this was an impurity that was formed by a non-complete conversion, potential non-complete conversion of an intermediate in one of the steps. And so from the theoretical assessment resulted that it was exceeding this purging factor. And that's so we did the spiking experiment, and it resulted that the process was not capable at that moment to effectively purge the impurity. So it was a real risk to have it in the final drug substance. So this is an example on how, without a risk assessment, you will never know that this impurity could form, no? And maybe you end up, at a certain point to discover this impurity in your routine, quality control, no, with a lot of challenges in terms of regulatory challenges, and also, of course, you can expose patients to safety risk. And so with the risk assessment, you can avoid the risk with this process, because now we have identified the risk, and then we could also take corrective actions to mitigate the risk. We have optimized the process parameter or the synthesis condition in the steps where the impurity was formed. Then we also set up a spec in the subsequent intermediate to really control the level of this compound. And with this mitigation, basically, we have excluded any risk of having the final, the impurity in the final API without even putting a spec in the API. So this is just to show you that the risk assessment is a theoretical exercise, but it has really some very practice consequence that can be very, very important. Okay, in the next slide, so I'm going to now talk about this challenge for the drug product, which is also very important, regarding the development of the dissolution method, and in particular, the challenge of the discriminatory power, because we all know that the dissolution method, the main function is to make sure that your routine commercial batches are representative of the clinical or bio batches that you have used to test safety and efficacy. And so this is a very important point, because really it could be a potential safety risk. So authorities are posing a lot of attention in making sure that the dissolution method is sufficiently discriminatory. And if you are questioned on this during the registration procedure, then it could pose a really serious risk, because it's not easy to change the dissolution method late in development or even during registration. And even if you succeed, then you have invalidated all the results that you have produced, and not only batch analysis or stability, but also the bridging studies that you may have performed to bridge formulation. So it's really a situation that could cause even a delay to time to market. And so it's crucial that this is achieved, and how the CMC can support by applying the QBD approach to the method development. because you know the critical quality attributes of your drug product, and then you can intentionally produce some drug product batches with those critical quality attributes to vary outside the acceptance criteria intentionally, and then you test those batches with a dissolution method, and then you prove that your method is able to discriminate, to reject those batches. And if you produce a sufficient number of data in this direction, then you are sufficiently sure that your discriminatory power is proved, and so you avoid such a critical risk during registration. So a very important support that is given by CMC in these cases. And if you go to the next slide, this brings me to the connection to the last challenge. This is a selection of batches and the associated bridging strategy, because you know that the batches that are supporting the registration, the data, I mean, that supporting your registration are not always produced with commercial batches, but often it's very common that are produced with development batches that may differ in terms of manufacturing process, in terms of batch size, even in terms of formulation. And so it's really fundamental to have a proper bridging strategy to bridge those batches and be sure they're representative. And yeah, it is obvious as this is a critical point, because if you fail in this, then only your development story is posed at risk, and so you could have really serious challenges during the registrations. And so basically, how can you prove that your bridge strategy is strong? But you had two solutions, or you run a clinical study, so you run a clinical bioequivalent study with bridge the formulations. And so in case of positive results, are super sure that your batches are representative. But of course, it's not easy in terms of money, in terms of time. It's really a challenge to run a clinical studies for every bridging in your development. And so we have also alternative, and this is where the CMC steps in to provide this alternative and to perform a sort of case-by-case evaluation to identify which is the better in vitro method that can be used to weigh the clinical study to bridge the formulations. And this is a very important support, because the CMC, knowing the type of molecule form, can decide which is the appropriate vitro study that can be used for the bio waiver of the clinical study. And so this is very important in terms of money and the time saving, and to avoid critical failures during the registration. Okay, in the next slide, so I had talked on how important is the CMC role to overcome these challenges. And now let me also spend some words to introduce some of the innovations that we could have in our work and will shape really the future of our work. So in the next slide, we can start from the artificial intelligence in CMC, because you know, of course, that AI is spreading everywhere nowadays, and it is already interesting our work, but it will be even more in the near future. So in PLG, we are starting to see, especially in big clients, but also internally in PLG, we are starting to implement solutions really AI driven. And I can provide a couple of examples. For example, systems that can pre-populate module 3 sections starting from module 3 template by obtaining information from source documents and from the applicable ICH guidelines to pre-populate those module 3 templates. And then the CMC author can use those files as a starting point. Or then I saw the same situation where AI did the same job for response documents. So to pre-populate response document templates by taking the questions from the deficiency letters, and then also information from the submitted module 3 from the applicable guidelines, and then to create a sort of first draft of response, and then can be used as a starting point. Then AI can be used to streamline the QC process of your dossier, sort of audit to find typographical errors, formatting errors, so they can facilitate this kind of work, or also could create a repository where the quality information are retrieved by the web, from the applicable quality guidelines, and then this AI system maintain the repository updated automatically by maintaining this information up to date. So this is very, very important. And so in case you want to implement some of these really quality of life improvements in your daily routine, then you can really rely on PLG and ask for our support, because we have the expertise really to support you in this journey. And the next one, it's for next slide, please. Yeah. Continuous manufacturing real-time release test. It is also very important because it's an innovative approach to manufacturing and testing the medicinal product, where all the operations are performed in continuous, and so you don't have any interruption, and also the unit operation are integrated into the system, and even the in-process control or the release of the product is in continuous. So it is obvious, as you have a lot of savings in terms of time and also in terms of waste, and also improve the productivity, but also the quality of the final product. Let me say that also it includes a lot of challenges and complexity also for CMC, not only for manufacturing, because it's not easy to reflect all this info in the module 3. So I'm not an expert on the field, so I will not go into the detail, but just to transfer the message that we do have a lot of expertise in PLG for this. We are already supporting clients in this. And so you can definitely, if you want to implement this challenging but rewarding new approach, you can rely on us, that we can really provide support in this. Okay, so I think it's all for me. I can give you back the floor, Rania, for the conclusions.
Yes, I think I was talking to myself for a second. So just to kind of summarize it and close it out, and thank you, Fabio. This was great. These were great examples that you've provided, and really provided real life examples of what we go through almost daily. So I would say that we talked a lot about risk assessments, right? So document risk assessments and the rationale for decisions throughout development. The rationale for key CMC decisions is truly the cornerstone of the story that need to be conveyed to the health authority. Don't underestimate the importance of a cohesive CMC story. You could have a great data, great product, but if you don't tell it right, it really defeats the purpose. So it's really important to have a coherent CMC story and how that aligns and feeds into the overall regulatory strategy. When you're providing the information, when you're writing, the information, it's critical to not only provide the required information in the submission, but what is the conclusion? What does it mean? How are you justifying it? What is your risk assessment for it? How do you mitigate it? It's equally important not to over-provide. Sometimes sponsors run into, we did 1, 2, 3, 4, so we're going to put everything in the module 3. You can keep a lot of it, a lot of your data in your internal documents. You have to only tell the story that is relevant to your final product. So any poorly explained data with inadequate justification can muddy your story. It can make the review more time consuming for the health authority. And truly the last thing any applicant wants is an irritated reviewer. So I would just say continue to be very transparent, have your frequent communication with the regulatory authority, think of your story, implement your QBD from the outset, and then just keep an eye on the regulatory landscape, and the guidance is constantly evolving, stay up to date, and it's never really a black or a white. There is a gray. It's meant to be flexible. It's meant to be not one size fits all. So it's your job to know exactly how to apply it and how to interpret it.
That was great. Thank you very much, Rania, for sort of summary statements at the end and giving a bit of a closeout and conclusion and your perspective. And thank you also, Fabio, for your contribution. So that brings us nicely onto the Q&A session. We've maybe got 5 or 6 minutes before we start to formally wrap up this session. So I'm just going to go into the chat box and see if there are any questions, and we'll pick 2 or 3 of those questions for our expert panelists to sort of go through. So bear with me while I have a quick survey of those questions, and there are a few common themes, but 1 or 2 I can pick out. So let me go. Okay, here's one. Yeah. During the pre-marketing application, then, what are the examples of common questions sponsors frequently find themselves asking and the reviewer or reviewers simply won't answer directly? I think this is probably a nice one for yourself, Rania, to step into. So I'll hand the microphone over to you.
Sure. So thank you, James. So I think one of the things, honestly, that I see a lot being on both sides, the sponsor side and then, the consulting side, is what level of the data is sufficient for submission. It's, FDA cannot give exact answers on that. The what level of data is sufficient for submission is probably the most common question for CMC, whether it's the amount of stability data, the number of validation batches, the depth of, like the impurity characterization. FDA will rarely provide a specific answer beyond the standard that they would, just copy and paste. This is the guideline, etc. This is what you need to do. And really, the reason lies in it's a case-by-case scenario. And so the agency is reluctant to endorse, a minimal approach without having a full context. The regulatory guidance provide considerable room for interpretation. It's not a one-size-all, and the FDA expects that. Well, it's FDA, EMA really is the health authority, but they expect that you would use scientific judgment and again risk assessment to decide how much data is enough and justify that decision. So let's take an example: the stability data that comes on the mind of a lot of companies, right? Especially if again we're dealing with fast track or orphan drugs. So small molecule, for example, the ICH and the guidances outline a standard data set for new chemical entity. It's typically you have 3 primary stability batches with 12 months long-term, 6 months accelerated. This is it. This is what it says. So the guidelines set a baseline, but it's not hard and fast rule. In real world, there are constraints, right? There is that, again, we go back to the aggressive timeline, the breakthrough therapies. So this means that sponsors sometimes will need to file with less, maybe 6 months of data, right? So will the FDA accept the 6 months instead of 12 months? Don't expect the reviewer to say yes or no in advance. The agency stance on that is follow the ICH unless otherwise justified. So if a sponsor asks this question in a, let's say, pre-MA meeting or pre-NDA meeting, the regulator will likely respond with include sufficient data to demonstrate product stability throughout its shelf life. In other words, the onus is on the sponsor to decide, is their data package supportive of the proposed expiry, and what is the risk they're willing to take. So telling the sponsor exactly, how many batches in an example like that is enough could actually backfire because, if the agency blessed the minimal data set in advance and then there's issues later on, that could compromise their standards. So here is where the sponsor is left with, guideline and, their own scientific diligence. So I would say if you come across something like that, and I myself have come across it on both sides, strategic advice is, when facing data sufficiency question, lean on good development practice, lean on risk management, right? Rather than trying to chase a magic number. Use your guidelines as baseline. I'm not saying ignore the guidelines at all. It's your minimum starting point. But if you must deviate, prepare strong justification, which would go obviously in your module 3, different sections within your module 3. Supported with stress data studies, for example, perform risk assessment. Don't wait until the end. While you're in phase 3, simulate commercial scale conditions early in development. Engage the agency early on, but ask the right question, right? So rather than asking, is this enough? Pose a specific question and say, we intend to submit with this on this number of batches. Does the agency have concern? Now, you may not get a yes or no, but you may get some glean whether the reviewer see any red flags. And so you could, use that to interpret and see, am I on the right path or no?
That was great. Thank you very much for your insights there. Rania, let's have a quick look at the time. Yeah, I think we can maybe get one or two more questions in. Let's have a look. So actually, I've got one here, Fabio, that I think you're probably well positioned to answer. So is the CMC approach applied during development affecting also post-approval lifecycle management? So yeah, I think one for you there.
Okay, yeah, it's a good question indeed. Yeah, let me, I would say shorter answer is definitely yes. And the long one, it's because the risk assessment provides a lot of flexibility. And so because with the risk assessment, you end up in defining all the connection between your process parameter, the characteristics of your molecules. And so the process really will allow you to not define, let's say, a certain threshold, really specific a certain threshold, but ranges as a certain threshold for your in-process control or for your specs. And so this is called, in the QBD, is called a design space. No, so are ranges of acceptability, not specific strict thresholds. Ranges where if you are into these ranges, you maintain the same level of quality. So this is the criteria of the design space. And if you achieve this, then this provides a lot of flexibility, even for the manufacturing or the quality control function in applying some variation during the life cycle, but still remaining in those ranges without any post-approval variations. So this is the main point, that it is an investment, the QBT, or in general the risk assessment approach, that the company can decide to do, of course, to streamline the registration process, because we have talked on how much it is important, but also it will provide an added value, and even to gain flexibility for post-approval variation for life cycle. Or you can even think that maybe if you have the product approved in Europe or in US, and you want to also start a new registration in another country, you could be questioned on the approved limits or on the approved information by another health authority. By having this risk assessment on your hands, you have a lot more understanding and information and package to use it to justify responses in the response documents, and so streamlining even that the process as well. Yeah, I hope it answers the question.
No, that was great, Fabio. Many thanks for your insights on there. Very conscious on the time as well. We've got about 2 minutes left. I'm just gonna get to the next slide, which is really my closing remarks, and I will keep it brief. But really, thank you again to our expert panelists there. Really thorough and comprehensive presentation around the CMC development of new small molecule products. We went for an overview of some of the pathways there that you might encounter in CMC development, some key considerations also with some examples, case studies, and associated challenges. So I think that was a really rounded view on this. I really like the future perspective as well on how AI is certainly influencing the way that we work, and it's definitely becoming more and more a part of a regulator's partner now. We're partnering with AI technologies there on a daily basis, and you heard from Fabio there that us as well as other larger clients are working there as well. So thank you very much for your responses, some of your questions and the responses provided. Any that we couldn't answer today, we will follow up with those at future time. So thank you. I'll now hand over to Gail for the closing of the meeting. So thank you, Gail.
Great. Thanks. Thank you, panelists. Thank you, everybody who joined us. This concludes our programming. If you have any further questions around today's presentation, please visit our website at productlifegroup.com. And for those who did register, we will follow up within a short period of time with a key takeaways document. Thank you all, and we look forward to seeing you at our next webinar.
Thank you.
Thank you. Bye-bye.